The eLife Podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, welcome to episode 41 of the eLife Podcast, which is produced by The Naked Scientists. I'm Chris Smith. This month, scientists discover the first anti-aphrodisiac, how epilepsy forms in the first place, and should science cease its obsession with novelty? But first, fish are social creatures. They're capable of learning from one another, and some species also have a clear hierarchy. So I suppose it figures that they ought to be able to recognise one another's faces. Muyun Wang is at the University of Tokyo.
So Medaka fish is only about 2-3 centimeters long, and they live in the rice field in East Asia, and they are very popular pets in Japan. So almost all the Japanese kids will have them as pets. Recently, they become very popular molecular model. So we have a good number of molecular tool sets to edit their genome, and they are also very good for studying neuroscience. That's why we are interested in this small fish.
And in particular, what were you interested in studying this time?
So we observe like the female Medaka, they prefer the familiar male. During mating, the male will swim circle in front of the female. And if the female saw this male before, she will accept him quickly. But if it's an unfamiliar male, then the female will take longer time to accept this male. It seems they can actually identify different individuals. So even after we exchange the male after familiarization, so we put one pair of male and female together and then exchange the male. But the female were able to notice like this exchange. So this is not my boy anymore. So she will take longer time to accept him. So seems they can perform very strict individual recognition.
Do you think they're doing that by visual processing? They're looking at each other, or might they be using other cues, for instance, smell, or perhaps a combination of those things?
Yes, so the using of odor cue is very common in fish because odor cue can spread more widely in the water. But in this case, we try to use visual cue, odor cue, and both cues in the fish. And we found with only visual cue, they are able to accept the familiar male very quickly. So probably they use only visual cue for individual recognition, and odor cue maybe is not so important in this case.
How did you physically do the experiment?
So when we put the male in the transparent cup and visual familiarize them for more than 5 hours, and then put them together for mating test. And also we try to condition them with electric shock. So we use a white maze. When the female stay at like the safe male side, then she will not get punishment. But when she swims to the wrong male side, she will receive electric shock. And we found after like 40 trials of training, the female were able to stay at the safe male side. So with both mating test and electric shock test, we think the female Medaka fish were able to discriminate different males.
In other words, in order not to get an electric shock, the female has to recognise which of the males she's seen before, which is the safe male, so she swims the right way. So they're clearly visually recognising the males that they like.
Yes, and also we are wondering which body parts they use for discriminating different individuals. So we try to cover their face and cover their tails. And we found the signal around the face is very important for individual recognition. So when you cover the head, then the female cannot recognize the male anymore. But when we cover only the tail, then they are still able to recognize this male. we also tried face inversion effect. So in human, faces is very special. So we recognize the faces as a whole. So not only with the like with eyes or nose or mouth. So we look at the face as a whole. So if the face is upside down, we cannot recognize the face so fast. So this is because we have a special brain part for processing faces, and when the face is upside down, the process become different. So we also test this in Medaka fish. So we use prism to invert the Medaka faces, and we found after inverted, the female also cannot recognize the same male. So I think it's comparable to the human face inversion effect. So maybe face is also processed specifically in their brain.
Muyin Wang. So who knows, maybe your pet Medaka fish can also recognise its owner. Some insect species are polyandrous. That means they mate more than once and with multiple males. But if a female has already recently mated, it's in no one's interest for her to mate again too soon, because sperm from different males would end up competing. So western tarnished plant bug males, and these are pests that devour strawberries and cotton crops, also transfer to the females when they mate anti-aphrodisiac chemicals. One is called myristal acetate, and another is geranyl acetate. These temporarily warn other males to steer clear. But now Colin Brent has found that the females use an anti-aphrodisiac to neutralise the effect of the anti-aphrodisiac so that they don't end up off limits for too long.
The male has these very large accessory glands within its abdomen. They're each about the same size as the guts within those abdomens. So they're very substantial organs, and they transfer a lot of constituents into the female during the mating. And then these are slowly released into the female or externalized from the female. And so some of these chemicals control her behavior. They'll actually reduce her interest in remating. And other chemicals are being volatilized out into the world. And new males will come along and attempt to court that female, but if they detect the presence of some of these odors, in particular myristal acetate, it serves as an anti-aphrodisiac. The males quickly lose any interest in courting those females.
Is the reason for that were they to court those females, their sperm would end up in competition with whoever's already mated with that female, but also it would impair the ability of the female to get on with the job of laying fertilized eggs?
Yes, so it negatively impacts both the female and the male, but it also has a positive impact. So the males benefit by not wasting a tremendous amount of time trying to court a female, which will quite likely not be receptive to their advances. Once the female's mated, there's usually a period of several days where she loses all interest in mating again while she's busy trying to oviposit. So if a male is constantly coming along, harassing her, she loses out on opportunities to deposit her eggs, and also the enhanced activity around that female heightens the chances that predators will notice her. The same thing for the males. So it's to their mutual benefit to have a signaling system that says this isn't really a viable mating opportunity.
Equally though, and I can see the merit in that, is there not a danger that the males could oversupply some of these anti-aphrodisiac chemicals so that the female loses interest but actually regains her ability to reproduce again before she regains her interest, and therefore a mating and a reproductive opportunity is potentially lost?
Yes, and that was one of the questions that sort of drove this research, in addition to trying to find mechanisms that we can use against these pest populations. We were interested in why this chemical system would evolve this particular way. If a male is recently mated, he'll actually transfer relatively little of this anti-aphrodisiac to the female, so she'll become essentially attractive again to courting males well before the time when she wants to be courted. On the other hand, if a male hasn't mated in a long time, has built up a great reserve of these anti-aphrodisiacs and transfers that to the female, she may have her capability of reproducing again or mating again masked by this excessive quantity of anti-aphrodisiac. So it would behoove them to have a signaling system that wasn't just sort of a one-note system that basically was yes or no, and having a quantity that varied from animal to animal. It basically makes it unpredictable and unusable.
And is that what they've got? Is that what you found?
No, in fact, what we found was that the female takes one of the chemicals delivered by the male, geranol acetate, and converts it over to a third chemical, geraniol. And this geraniol can actually serve to counteract the anti-aphrodisiac effect. So as the chemical gets converted over the course of time, there's more and more of this geraniol and decreasing amounts of geranol acetate and slowly decreasing amounts of myristyl acetate. And that geraniol actually counteracts the negative effects on the female's attractiveness. And we can topically apply a mixture of these chemicals to the female and render her attractive or unattractive depending on what ratios we use.
So summarising that then, the male puts into the female myristyl acetate, and that's an anti-aphrodisiac. He also transfers geranol acetate, and that together with myristyl acetate is an anti-aphrodisiac. But at the same time, that geranol acetate is being converted into geraniol, and that actually kills off some of the anti-aphrodisiac effects. So you must therefore have an anti-aphrodisiac effect. Is that reasonable to say?
Yes, it is. So essentially, instead of having a system in which the absolute quantity determines how males will respond to the female's odor, you have a system in which there's greater balance where the proportion of one is going to be dictated by the proportion of the other. And it's measuring the relative proportions that gives the males a greater ability to accurately assess the female's reproductive state.
And is this exclusive to this particular group of insects, or do you think other polyandrous insects that mate with multiple male partners, do you think they're using a similar or even the same chemicals?
We've looked at a number of different species, and we found that there are several that have these anti-aphrodisiacs. This is the first instance in which we've actually found an anti-aphrodisiac. There is evidence in Drosophila, the fruit fly, that the females will actively expel the anti-aphrodisiacs that the males are delivering to them during mating. And so at least that one species has this active process of countering the negative impacts that the males are having from this passive mate guarding system. I suspect, however, that because we didn't know to look for it before, that if we were to actually go through and examine a number of these species where anti-aphrodisiacs have been found, we very well may find that other chemical anti-aphrodisiacs exist.
Isn't nature an amazing thing? Colin Brent there, he's from the Arid Land Agricultural Research Centre, Maricopa. You're listening to the eLife podcast with me, Chris Smith, and on the way, is science putting too much emphasis on breadth at the cost of depth? But before that, about 1% of the adult population are affected by the seizure disorder epilepsy. This happens when a small cluster of nerve cells in a particular part of the brain become electrically unstable. This abnormal firing pattern can periodically spill over into other brain regions and then cause the disabling symptoms that patients display. But what causes this so-called epileptic focus to form in the first place, and how that causes the changes in the surrounding brain tissue which eventually result in seizures, wasn't known. Now we have some new insights. Carola Haas.
The approach we used was to mimic the disease in mice. We can inject a toxin and then observe structural changes occurring similar to the situation in humans. Disease progression, so cell death and structural reorganization of the brain network until the point that seizures emerge. And we used magnetic resonance imaging to study what's going on after injection of the toxin in the mice brain. And we could do that for a period of 3 weeks. And this gives us information about structural changes, but also molecular and metabolic changes.
Right, so just to recap then, you've got young mice, you expose them to a toxin which causes damage to the right part of the brain so that these animals within about 3 weeks develop a clinical syndrome of seizures, a bit like what would happen to a human, and you're able then to use imaging to follow how that state evolves in these animals. So you can see what changes happen in the brain upstream of the fully fledged clinical condition occurring. So we can get some insights into what might be happening in the brain of a human who is going to develop these sorts of seizures.
That's a reasonable summary. But also what I would like to point out is that we can study this in periods of weeks, which is first of all not possible in humans because it takes many years, and we also cannot put humans in the scanner over many years. So that was our idea, that we try to find early indications of later disease progression.
And now, having made these observations, what changes are happening that appears to predate the emergence of the epilepsy? And could they be used as a useful predictor in humans, do you think?
First, I would like to say that we found very early changes, so within 24 hours, which reflect the death of neurons, and this predicts the severity of the overall structural changes referred to as sclerosis. And this sclerosis is also something which we find in humans which are epileptic. So the early changes already predict how severe the sclerosis will develop at the end of the disease. Also, neurotransmitters were severely changed. Excitatory transmitters and also inhibitory neurotransmission went down. If we looked at a little later, we saw that in other areas, microstructural changes were occurring, in particular cell types, which we hadn't expected before. So not only neurons were affected, but also glial cells were severely changed in their structure. And these changes were also important to predict the severity of the disease outcome at the end.
Are we only able to diagnose people a bit earlier and just tell them you're going to get epilepsy earlier? Or does knowing that a person is at risk of this happening from an early stage give us an alternative therapeutic opportunity if we intervene with treatments before the person overtly develops the seizures? Because the seizures can make themselves become worse in the long term, does that mean we have an opportunity to potentially reduce their severity or even stop them altogether if we start therapy sooner?
Exactly. That's the big outcome of this study, that we have hopefully tools that we can test new anti-epileptic substances which are not available at the moment, because the substances which are available at the moment, they only treat the symptoms, but they don't cure the epilepsy. So the big hope we have is that we can use our study to find new medication.
Let's hope so. Carola Haas there. She's at the University of Freiburg. Each month in the eLife podcast, we devote some time to trying to look beyond just the results of scientific endeavour and to consider also the practice and some of the societal aspects of science. Previously, we've heard about diversity in the discipline, how women and early career scientists are faring in the modern scientific era, and how as a community we need to be careful to avoid distorting the facts. This time, Washington University's Barak Cohen has something he needs to get off his chest.
There are a bunch of reasons I wrote this. First, the accumulating number of rejection letters that I was getting for both my grants and papers, which were stamped as being not novel enough. So that's just to admit to you that I'm a human being and fallible. But the other reason that I wrote the piece is that I'm becoming more and more interested in issues around the philosophy of science, and in particular issues about why science seems to work so amazingly well, especially relative to other human endeavors.
Let's talk about these rejection letters first, because Let's get that out of the way. What is the scale of that problem in the US and also beyond the US, worldwide?
Well, my impression is that it's large. I mean, if I'm representative of a typical scientist, then I would say probably at least half of the rejection letters I get coming back are lack of novelty as part of the reason. In terms of grant proposals, I would say it's got to be at least in the 50% range. And I see that from the other side because, of course, as a working scientist, I not only submit grants and papers, I also have to review papers and grants. And when I'm sitting in the room on a review panel, the review panel talks a lot about novelty, and novelty is a big reason that grant proposals get rejected these days.
But why shouldn't we be striving for novelty? Because isn't that partly why we're all in business? We want to know how the world works. We want to discover new things. So it is important that we prioritise that.
Yes, and science has been amazingly good at uncovering novel phenomenon. If you just look at the pace at which new discoveries are being made in molecular biology and have been being made in molecular biology over the last 50 to 60 years, the pace of novel discovery is incredible. And that is without an explicit emphasis on doing novel research. I guess part of what my piece is about is that there really isn't a need to explicitly reward proposals that are novel. Novel lines of research are important, but it is also important to explore things deeply. So with a finite amount of effort, you have to apportion that effort in ways that both open up novel lines of research and encourage people to probe deeply into those phenomenon.
Do you not think that there might be a historical bias here, in the sense that a lot of the novelty was novel because it was easy to stumble upon, relatively speaking, and that now we're in that law of diminishing returns where, the curve has begun to flatten and we're having to invest more and more and more to fill in the gaps because the big landmark leaps have been made?
Always a dangerous thing to say, but I do sort of think that when a field is young, and molecular biology and molecular genetics is still a pretty young field. In the beginning, there are lots of foundational principles to uncover, and big discoveries are made quickly and rapidly. And then you do get to a position of diminishing returns. If you're thinking about it that way, if you're thinking that, in order to be successful, I have to uncover a foundational principle. And I guess part of what I'd like to argue here is that you can be a very successful molecular biologist by taking principles that we qualitatively know about already and putting them in a quantitative framework that makes them predictive and useful.
Well, go on, explain what you mean by a quantitative model then. Give me a tangible example of how that would apply to molecular biology.
One of the things that I work on in my own lab is transcriptional regulation. If you ask me, how does a gene get turned on and off at the right time in the right place, the language that I'm going to be using is mostly qualitative. And so it's very difficult to take that qualitative understanding and turn it into a quantitative prediction.
Do you think that we need to be prescriptive about that? Or will it not just happen anyway? Because as people are opening up new avenues and discovering new things, they will gently fill in the gaps behind.
I think we do have to be prescriptive and encourage people to think about quantitative models because it does not seem to happen naturally in molecular biology. My advisor used to tell me that, you can conquer the world as long as you have an internal control. You can always tell where there's a difference. But what was missing in that statement is that it might matter whether it's a twofold difference or a tenfold difference. And how we incorporate those sorts of thinking into model building, I don't think that really comes naturally to molecular biologists, and particularly in the types of training that graduate students go through during the PhD.
They're not taught much about philosophy either, and there's a big chunk of that in the article you've written. I found it quite refreshing.
Yeah, well, I think maybe there's a reason the degree that we get is called Doctor of Philosophy. It's not actually called Doctor of Biology or Doctor of Chemistry. It's called Doctor of Philosophy. And I think the reason for that is that the really important skills that graduate students learn as they go through the PhD are not actually how to do a PCR reaction or how to write a particular piece of code. It's how to break down a problem and break it into manageable pieces, and how to know when you've collected data that really answers that problem, and how to synthesize data into a coherent argument. And those are really the important skills that you learn as a PhD student, and I would say that, those come straight from philosophy.
Barak Cohen. And on that thought-provoking note, there we must leave it for this month. You've been listening to the eLife podcast, previous editions of which are also on eLife's website, that's elifesciences.org, or you can subscribe on iTunes. This is the Naked Scientist production. I'm Dr. Chris Smith, and you can find out more about what we do on our website, that's at nakedscientist.com. I'll be back with another look inside eLife next month when we'll be celebrating 5 years since eLife first began. Until then, goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.